Hi, I am delighted to present our paper Cautious Optimism in Safe Dose Finding Clinical Trials. We develop an adaptive clinical trial method with drug combinations, to find the dose combination, named MTD, whose estimated toxicity is closest to the target. Also, for the safety of participants, in each trial, the average toxicity of the trial should not exceed the target threshold. The major challenge in dose-finding clinical trials with drug combination is the conflict between the exponentially increasing number of DCs and the limited patient budget. To address this, we use a joint parametric dose toxicity model that represents the toxicity probabilities with a few parameters. Then, we can estimate the toxicities of all the DCs by learning the parameters only. Based on the model, we proposed an adaptive Bayesian safe dose-finding clinical trial design named SDF-Bayes. In the design, the parameters are learned by the Bayesian approach and the DC to be tested in each round is chosen based on them. Now we describe the main ideas of SDF-Bayes. First is optimism for efficiency. To choose the DC to be tested, our method assesses the most likely DC to be the MTD. For this, as shown in the figure, we use the probability that the DC belongs to the targeted interval based on the posterior distribution of the parameters. Then, we choose the most likely DC. At the same time, we consider a principle named cautious for safety. To guarantee safety, we conservatively assess the possibility of safety violation for each DC by using the posterior distribution. If the chosen DC is unlikely to violate the safety constraint, then we keep the choice. If not, we discard the choice and choose the unlikely DC to violate the safety constraint. By this cautiously optimistic procedure, we can find the MTD more effectively and maintain the safety constraint. In our paper, we also consider heterogeneous groups. We consider M-identified groups and the total budget of T patients. The patients should be adaptively recruited across the groups to find the MTD of each group. For adaptive patient recruitment, we estimate the expected improvement of the probability of the most likely DC to be the MTD assuming the occurrence of a toxicity event. By recruiting a patient from the group with the largest expected improvement, we can recruit the most informative patient. Finally, we evaluate our SDF-Bayes by comparing its performance with state-of-the-art adaptive clinical trial designs. For this, we mainly use 3 synthetic datasets and a real-world dataset. From the table, we can see that SDF-Bayes makes fewer errors than the other designs in most cases while satisfying the safety constraint. For heterogeneous groups, we use the groups A, B whose toxicities are given by synthetic A and B, respectively. And we allow for a total of 80 patients and adopt adaptive recruitment to SDF-Bayes and SOTA-Bayes. From the table, we can see that adaptive recruitment improves them. For more information, please visit our website. Thank you for watching.